Good morning, everybody. Anybody got questions while we wait? Questions. Ba-doo-ba-doo. Doo-doo-be-doo-be-doo-be-doo. Okay, I'm gonna get going anyway because people are here, and if they're not, they're not. So, set you some homework in a sense. Last class, which was to do this exercise, right? So, hang on, share screen. All right, this exercise. How do we use this to figure out the relationship between these four genes? Where do you start first? Okay, give you a hint. What do you think 50% recombination frequency means? If we go up to, dang it, where is it? If we have recombination occurring in 50% of the chromosomes, what does that mean? Yeah, they're far away from each other. They're not linked, right? We can't tell how far away they are because you can't get above 50% recombination frequency. That's like that's the hard ceiling, right? So whether they're twice as far away from each other or 4 times as far away, it doesn't matter. They're still going to have recombination occurring every time between them. So if A is so far away that it's not linked to B, and it's so far away from C, it's not linked to C, and it's not linked to D, right? So where would we put A relative to B, C, and D? They're going to be close. It's going to be far. Yeah, it's going to be far away, right? So now we can build a map like so. Stick that over there, right? Now we're going to have A, and I'll just do it from left to right. It doesn't have to be, right? And we're just going to put them as B, C, D over here, right? Because this is 50. Make sense? Now, how do you sort out the relationship between these? Which gene pair is the closest together? B and D, right? So we have A and we have B and D. That's 10, right? The numbers are the recombination frequency, which essentially is the distance, right? Okay, what's the next closest together? Yeah, B and C, right? So now we have an issue, right? Is it this or is it this? Which one of these different combinations is correct? What's the next piece of information that you need to use? So if B and C are 20 and B and D are 10, C and D are 30, right? So one easy way of doing it is, well, what adds up to 30? It would be B and D would be 10, and this would be 20. Make sure you can see that. Ah, there you go. That's a bit better. All right, what's that? What here adds up to 30 in terms of distance? Is it this or is it that? Which one makes the most sense? CBD, exactly, because if C is 20 from B, C is going to be 30 from D. We look at the recombination frequencies, and that's what we get. So the, here it is. That's the order of these three genes. Right. Now, question. Is it this or is it this? Which is correct? Hint, hint. It's a trick question. Both of them. You know that the relative order of these three is CBD or DBC, right? But these three are so far away, I've got to put this in, from A, you have no idea whether C is closer to A or D is closer to A. So the best you can do is say it's going to be this or this, right? Does that make sense? Right. You're using distance and combination of distances to figure out how far apart things are. Okay, take a quick picture of that. Don't want to spend a huge ton of time on this because we've got 3-point crosses to do and they're way more complicated. Oh, crumbs. Okay. Any questions about that? Okay, so for 3-point crosses, right, essentially you can do these kinds of jobs as 2-point crosses. The downside is, or downsides, right, are that for one, you have to do a lot of crosses, right? And two, essentially you get kind of almost like rounding error inaccuracies, right? You're not looking at other recombination events at the same time, right? So you tend to underestimate underestimate distance, right? And that's because these double crossovers can go undetected, right? And so doing a 3-point cross can be a more accurate way of doing this. Also saves a bunch of time, right? And so you can do both distance and gene order in one cross, right? So you get a more accurate result, right? So I'm going to show you a kind of a picture of this, right? We're going to talk about this for a bit, and then we're going to, what the cat's doing? We're going to do a bunch of practices, right? Because this is how you get good at doing this stuff. It's just like doing math problems, right? Okay, so essentially what we're going to have now is we're going to have 3 different genes. Our individual is heterozygous for each of those 3, because otherwise you're not going to see any difference. And we're going to be looking at essentially, I have no idea what that cat is doing, 4 lots of progeny. All right, we're going to be looking at, we're going to be seeing parental, right, where the offspring inherit an intact or non-recombinant chromosome from their parent, right? So we're going to look for progeny that have this collection of alleles and have this collection of alleles, right? If linkage is occurring, which group, recombinant or non-recombinants, do you think will be most common? Which type of progeny are you going to see more of? Ones with parental chromosomes or ones with recombinant chromosomes? The other way around. Parental, right? Because if linkage is occurring, there's going to be an excess of parents, right? And a lower than expected number of recombinant progeny. All right. Obviously, because if linkage was not occurring amongst any of these 3 loci, then you'd always have recombination between them and it'd look like they're on different chromosomes, right? Yeah. So ones that look like this are going to be the most common, right? And this is important, right? The reason why I'm telling you this kind of in groups like this is that when you see a collection of data for a 3-point cross, you need to figure out quite quickly what's what, right? And this is how you do it. Okay, so most common, and then you're going to get 3 different types of recombinant. You're going to get ones which are recombinants between A and B. Okay, so these will show essentially a difference from the parents in which of these 2 alleles you get together, right? So in the parents, it's dominant, recessive. In the recombinant offspring, it'll be dominant, recessive, or recessive, dominant. Right? So the only change that is occurring is between these 2 alleles. Okay. Now you get another type of single crossover, right? These are going to be between B and C, and the same applies here, right? Everything will be the same, A and B, but in the recombinant, B's and C's will be swapped. You'll get a different combination from what you see in the parent. Those are the two single crossover events. Two single crossover. There you go. Separate fingers. Now, these are not necessarily going to be present at the same amount. You could have a map that's like that, where these two genes are really close together and these two genes are far apart, but still linked. You could have it even, you could have anything like that, right? You've got no way of knowing until you actually do the cross and collect the data. Now, if you have a certain probability of recombination between these two and a certain probability of recombination between these two, what is the probability that you'll get a recombination event between both of them? Think about your independent event rule. Because these recombination events are independent of each other and they're random. And so you're asking, how often do I get two independent events? What's the rule for independent events? Do you add the probabilities together or do you multiply them? You multiply, right? So if you get a probability of a quarter here and a tenth here, it's going to be a quarter times a tenth, whatever that is. Smaller number, right? So I shouldn't use that. I should use tenth times a tenth. Okay, anyway, doesn't matter. So these double crossovers are going to be the rarest group of all. So whenever you look at your sheet of data, and I've got one worked out just here for you, did that yesterday evening. Oh, there's my wife. Then you need to look for what's the smallest number of offspring. Those two lots, because you obviously got the reciprocals, right, are the double recombinants. Okay, so essentially, when you do a 3-point cross, I'm going to write out a Matt's guide to 3-point crosses, right? And as long as you follow this, it's really straightforward, super simple. Hello. Okay, first step on your path to wisdom and happiness is find out what are the fewest progeny, I guess. Right. So you're going to have, there will be two groups for each class, which is parental single one and single two. Or actually, let's use letters. That'd be better. Single A and single B. And double AB, essentially, if A and B are where recombinations are occurring, maybe. Hopefully that makes sense. Kind of, it's hard to type this stuff out and do so. Okay, so there'll be two groups for each class. You're going to have essentially the reciprocals, right? So if you have an individual which has, see, love, crossover between A and B, right? You're going to have dominant A, lowercase bc. But because it's reciprocal, right, you have to have the mirror image. It's just how it works. You're going to have lowercase a, capital BC, or dominant B and C, right? Hopefully that makes sense, right? So for every one of these four classes, you're going to have two groups of progeny, right, which are going to be the mirror image of each other, right? Okay. Anyway, these are the double recombinants. Okay, the key to these, I mean, they're important in another way too, but a little bit less so. But the main importance of the double crossovers is they tell you gene order. Because in double recombinance, the one that moves is the one in the middle. Okay, so in this case, ABC, super simple, right? B is the one that's moving, right? Because if you look at the recombinants, they are the same as the parents for A and C. And so the only way you could get B being different is if B is in the middle. I want to let that percolate in for a little bit because that is super important. Does anybody have any questions about that? Just for emphasis, super important. And I will not necessarily give you the gene order in the correct order when I give you a cross. So you cannot assume, right? And I'll write that down again for posterity's sake. More capital letters. I'm not shouting, honest. You can't assume that the gene order you're given is the correct one. You have to work it out from the double recombinants. Okay, easy, right? Then step 3, work out distances. For crossover 1 or A, 1A, whatever you want to call it, that is add the single crossovers one or A or whatever, the first one, right? Whatever you want to call that. And the double crossovers. And that's exactly the same. I'm just going to cheat and copy that. And that's it. Super simple, really. It looks crazy complicated. And it may be a little on the intimidating side because it's going to look like this. But as long as you follow that plan and that rule, you cannot go wrong. Right? It's like doing a chi-squared test or dihybrid cross or any of those other things. If you follow that turtle approach to doing things thoroughly and slowly, you can't get it wrong. However, if you want to do it faster than the turtle, you need to practice, right? And you will get questions on this in the exam, guaranteed. We're spending 3 lectures on it. So this is big stuff, right? And you need to be able to do these things like that. You can't spend 10, 15 minutes trying to figure it out because that will screw you for the rest of the exam, right? So you know you need to practice this stuff because you're time limited in the exam, right? That's the downside of having an open book exam, right? Well, I mean, all exams are time limited anyway. I mean, that's just kind of the nature of them typically. But it's even more so because I know that you can just look up stuff on the internet. So, you know, it's going to be a little bit like so, right? Okay, so this is what a 3-point cross would look like. Okay, you're going to have a cross at the top, which is telling you what is going on. Typically, you'll have a heterozygote, which is this one on the left, right? We're using capital letters and lowercase letters. We go all over the place in terms of notation. It is what it is. And we have a homozygous recessive tester, right? So this is essentially a back cross, right? The same as what you're doing in lab with your flies. Sometimes this might be a male, right? If these genes are on the X chromosome, right? And the organism reproduces with a XXXY kind of deal. But it doesn't matter. These ones here do nothing, essentially, in terms of what you see. They simply allow you to see the occurrence of recombination events between these three different genes in this individual. Okay, and so the next thing, so this is where it gets a little tricky, and you'll need to use paper to do this, right? Which is why, again, I had to work this out on a piece of paper. We'll get to that in a second. You need to figure out what's moving, right? And you can only do that by looking at what the parental chromosomes look like. So one, you're going to find out which are the parents, right? So I'm going to ask you here, which are the parents of these, which are the parental chromosomes in these offspring, the non-recombinants? How do you find that out? I told you earlier up in slide 21, one above this. Which is going to be the group with the highest number of progeny if linkage is occurring. Again, don't worry about that. Yeah, the highest number of progeny, right? Yes, exactly. So the ones which have the largest number of progeny are have the non-recombinant parental chromosomes, right? That's going to be this one here, right? 286, and this one here, 272. We're not necessarily going to put them in a nice order for you, right? We might do if we're feeling kind. We might not. Doesn't really matter, right? Because you're looking for parental chromosome, right? So these are the non-recombinants, okay? Which are the double recombinants? Actually, it even tells you on the slide. Just want to kind of hammer this point home. Which are the double recombinants? The lowest. Okay, so now here is a question you're going to have to think about. So what is the correct gene order? Again, you've got to look at what is moving in the double recombinants, because the one that moves, the allele that moves, is the one in the middle. So you look at the parentals, the non-recombinants, you look at the double recombinants. The one that changes, the letter that moves, or the gene allele that moves, is the one in the middle. Nope. SU. So if you look very carefully here, one double recombinant is, I'll type it out so you can follow along on that too. Is LZ. Actually, I'm going to do this all in capitals, so make it even more obvious, right? So the parents are all dominant or all recessive, right? This is a cis configuration. So when you see two dominants together and a recessive, it's the recessive one that moves. Or if you see two recessives together, right, which is this one, right, and a dominant, it's the dominant one that has moved. So the gene order that you're given here is not the correct one. So the correct gene order is LZ SU GL, or because we don't actually know in terms of its orientation, GL SU LZ. Does that make sense? If it doesn't make sense, you're going to have problems. So I'd rather know now, so I can kind of help correct it or explain it. Yay or nay? Confusion and despair, or happiness and joy? What do we have? Or just crap, it's Friday. I don't have any brain power left. Probably that too. Most certainly is, which means it's Saturday tomorrow, which is awesome. We still have a little bit of Friday stuff to get through, unfortunately. Okay, it's never, yeah, I know. It's never like, oh, I mean, I could do this on a Monday morning, but that's not going to be any better. Actually, I don't think it matters what day of the week I do this on. It's still going to be tough to get your head around. Okay, so the one that moves is always the one that's in the middle when you're looking at double recombinants. Right, that's the very first thing you've got to work out. Otherwise, your whole map will be wrong, even if the distances are correct. Actually, the distances won't make sense either. If you remember back to the 2-point cross above, if you work out, BCD, and you got the order wrong, then the distances aren't going to make sense, and your map will be wrong. Okay, so you have to work out the gene order first using the double recombinants, which are the fewest that you find. Okay, then what you need to do is work out distances. Okay, so these are the parentals. We're not going to use these anymore because they're not important, right? What we now look at are when recombinations occur between two things, right? So now we actually have actually, I'm going to change this because this is just unnecessarily confusing. This is a hangover from previous person that taught this. Okay. So these are the parentals. This is now the correct gene order, right? You could do it the other way around, but we're not going to bother because it's needlessly complicates things, right? So LZSUGL, right? So the parents, dominant, 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 recessive, 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 great. don't care about those anymore. They just contribute to the total. Now what we're going to look at is, okay, the first recombination event between LZ and SU, right? That's going to result in being a dominant LZ and recessive the other two, or recessive LZ and dominant for the other two, right? So that are these ones here. Now those are the result of single crossover events between A and B. So this is LZ and SU in that example, right? However, you also get recombination events between A and B in double crossovers, or between LZ and SU in double crossovers, right? So you need to, add those in as well. And so if you want to work out recombination frequency or distance between LZ and SU, you have to, absolutely have to. This is where it becomes more accurate than a bunch of double or 2-point crosses. You have to add the double recombinants to the single recombinants for that crossover event. If you don't, you'll get it wrong, right? So now it's going to be these two numbers, right? Because these are the reciprocals of each other, and these two added together, right? So it's going to be 40 and 33 from the singles, and 4 and 2 for that, which is 79. And recombination frequency is always the number of recombinants over the total number of progeny, right? So that's 79 divided by 740. Over here, that gives us 10.7 centimorgans, right? Or percent, and percent is essentially centimorgans, right? And then we do the same for the second crossover event, and these are similar distance from each other, right? We do the same. We add the single recombinants. Again, these are reciprocals, so you have to add them together. And the double recombinants, because again, just to kind of hammer the point home, if you're looking at a single crossover event here, or you're looking at the recombination frequency between B and C, or what would it be, SU and GL, you have recombination occurring both here and here. So you have to add those together, and that gives you 109 over 740, which is 14.7 centimorgans. Okay, questions? All right, so I'm going to give you one. Remember where is it? Up here. Ooh, all the way up here. We have this one with diminutive males and white-eyed females. And we have this one, which is between white-eyed females and miniature wing males, right? So these are all on the same X chromosome. And I was like, well, it'd be kind of cool to figure out what a 3-point cross between them would look like. So I actually went to Flybase, right? This is DM, actually even had a fly in front of my screen then. Yeah, so this is diminutive males, yes. And I did the same for the other ones. So I actually found, hang on, let me just show you, kind of neat in a way, just before I forget. So this is the recombination distance, right? Just to show you what this kind of information looks like. This tells you, this one here, that it's on the X chromosome, right? This is actually what the gene kind of looks like, I guess. And this is actually its physical distance, right? So these are in millions of bases, right? So this gene spans from this, I'm not even going to read it out, to this in terms of physical location. In terms of recombination, it's 3. doesn't do it anywhere near as kind of fine-tuned as this does. But anyway. And you can just look that up yourself if you wish. You'll find the exact same information that I did. And so I used those numbers to work up a 3-point cross. Right, so this is with those 3 genes, right? So above you see that there's 2 in the PowerPoint. There's 2 single crosses, 2-point crosses. And what we're going to do is I'm going to give you a bunch of information, and we're going to work through this as a 3-point cross. Okay, so we're going to cross. I'm going to use plus and letter notation because I think that's a lot clearer than a whole bunch of letters. It becomes a bit of an alphabet soup otherwise. All right, so this is what we're going to be doing. We have a heterozygote. All the genes are in cis, so it's all dominant, 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 all recessive, recessive, recessive. This is a heterozygote. This is essentially a homozygous recessive. Kind of not, because there's a Y chromosome there, but effectively it'll behave as such. Okay, test to the class. Is this the correct gene order? Is that the correct gene order? Can you go, yep, that's definitely the order that they're present in on the chromosome? Well, it might be.
I'm still confused on how you found the correct gene order from the
From the doubles?
mhm.
Okay, so let's go through it and hopefully I'll answer your question, because these are kinds of things that are best done in terms of practice, right? Rather than theory. So keep that question in mind and see if I answer it as we go through this, right? If I don't, then we'll do it again, okay?
Okay, thanks.
Right, so this might be the correct gene order. It might not be. We don't know, right? That's why we're going to be doing this cross, right? And I've actually got to write all this stuff out. Okay, so what I'm going to do is give you essentially the chromosomes, right? Because these ones don't count, that are either parental or recombinant, right? So we have and I'll post this up to Blackboard afterwards. Just getting enough space. Fortunately, put the most important ones at the bottom. That adds up to 1,000. Oh, hey, sorry. I did say I put the most important ones at the bottom. Okay. All right, so you did this cross. You scored 1,000 flies, which is quite a feat of endurance. And you got the following combinations. Now, essentially, this is wild type for all phenotypes. This would be recessive for all. So these would be white-eyed small flies that have small wings. There. This would be red-eyed small males with small wings. This would be white-eyed normal males with normal wings, for example, right? So this is how you score all of these based on their combinations of phenotypes. Now, scam call this early. Yep. So I just have to stuff that down the back of the couch so it doesn't make too much noise. Okay, so which are the double recombinants? All right, shut up. I can get it out there before I forget where it is. Which are the double recombinants out of all of these? 3 and 2. These ones. Ooh, let's use a different color. Okay, these are the double recombinants. So let's do it up in this color. Okay, which thing moved out of these three things to create those double recombinants? Which one is different from our parentals? DM. So the only way that DM can move and that result be caused by a double recombination is if DM is where relative to these three?
In the middle.
In the middle, exactly. Awesome. So this gene order is wrong. It is actually plus DM plus W plus M. Right, so the gene order, that's terrible. So the gene order is, I got that in shade, never mind. W, D, M, and then M. Okay, because that results from, if we put this on a straight line here, that's what had to happen to be able to swap DM from this chromosome up to here and the plus from this chromosome down to here to create these two chromosomes. Jelani, did that help explain things?
Yes, that helped a lot.
Good. Yeah, this is something just like any practical skill you have to practice, right? To be able to actually really understand what's going on. Just talking about it doesn't always help. Oh, man. I don't know why my camera does that. Anyway, okay. Have you all made a note of this? We're going to be using this to do some calculations in a moment. Okay. And sometimes it actually helps to rewrite things down in the correct order, because then you can figure out what is actually happening, right? Because otherwise it can get a little bit confusing. So really we've got plus, 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 and W, D, M, M are our chromosomes. are single recombinants for these ones. They're going to be any ones where the W moved relative to the DM and the M. Right, so that's going to be ones which are plus DM M and W plus plus. Right, these ones are 6 and 4. And we have our double recombinants, which are plus DM plus and W plus M. These are 3 and 2. Okay, so essentially, single recombinants are these. Double recombinants are these. All good? Add those together. 15 divided by 1,000 equals 1.5% or 1.5 centimorgans. So this is the relatively straightforward part. Figuring out gene order is the hardest bit. And then you do the same for the other one. Right, I'm going to leave those up. Actually, I do need to leave that up there. Right, so now we care about single crossover events between DM and M. Right, so it's going to be plus plus M or WDM plus, and double recombination events, which we already have down here. Right, so single is going to be plus plus M or WDM plus plus M, that's 165, and the other one is 160. Okay, add all those together, you get 330. That gives you 33%, 33 centimorgans. Now, when you draw these up as a map, now it will look like so. And this is the X chromosome. We're going to have it look like this. So this is going to be 1.5. This is going to be 33 centimorgans. And this is about 34.5 centimorgans. And that's actually what data from Flybase. But it also matches very closely the data that you get from those two two-point crosses. Okay, so that's quite a lot to take in one go. I'll post these to Blackboard afterwards so you can follow through what we did and figure out, what we're doing at each step. use the order that I put in the chat box, because that's how I did it here. And that's, in my opinion, the most reliable way of doing it, right? Because you can't figure out where recombination's occurring without knowing the gene order, right? And you have to figure out the gene order first. And then, I know I'm going over a few minutes, but I'm nearly done. Then I want you to go on and do this one, right, as a practice, right? And automatically, you can tell which are the double recombinants, right? How can you tell? Because they're the ones with the fewest progeny. Yeah, exactly, right? So start with those, work out gene order, and these have even another different notation, right? Further confusing. Use whatever notation works most for you. This one, the plus and the letters, works for me because I think it's visually the simplest, right? Whatever works for you is great. And then work through this. And if we get time on Monday, what do we have on Monday? Bum, bum, bum. DNA structure. If we have time, we can go over it on Monday, right? Just to give you that extra practice. And there should be plenty of problems all over the place, your textbook, internet, whatever, where you can practice this. And this is really not very hard to do once you understand how to do it, like most things, right? And the key to understanding how to do it is just to practice. And once you do, it'll be a piece of cake. Okay, so practice that between now and Monday, and find as many other examples as you can to practice with too. And you'll be as good as I am at doing it, right? It's not like a magical mystery trick that only like the special people can do. Anybody can do this. You just have to follow through the steps. All right. Sorry for going over. I thought it was kind of important to finish that up before we separate. Have a fabulous weekend, everybody. I'll post all these materials up within ideally the next half hour. So you can work through those yourself, see where your understanding matches and it doesn't matches, and then figure out whatever you need to so you can do it. All right. Have a lovely weekend, everybody.